Good morning, students. Today we are going to start with Term 2 syllabus. The first chapter of Term 2 is Cell, the unit of life. Here in this chapter we are going to read about cell in detail and the component present inside it. Our first topic is what is a cell? You all are aware that biology is the study of living organisms. Here on this earth, different type of organisms are there, ranging from a small microscopic amoeba to huge whale. Various organisms are there. But what is common in those organisms? Each and every organism on this earth is having a common feature, which is a cell. Their body is made up of cell. So if we define cell, we can say it is a fundamental, structural and functional unit of all living organism. It is the smallest part of our body which can independently exist and can perform necessary functions of life. Every organ of our body, such as skin, brain, muscle, bone, every part is made up of thousands of such cells. Similarly, in plant parts like leaf, root, even wood, huge number of cells are present. That means every cell has its own life. Old and weak cell in body, they continually die and replaced by the new one. This means every cell grow, then divide, afterwards it become old and then die. Then this is replaced by new cells. Here an example is given of RBC, human red blood cells. They have life of 120 days. After every 120 days, red blood cells die and new red blood cells are there. They replace the old ones. So all living organism, including human being, every organism start their life as a single cell, which is known as zygote. You can underline this. Cells are so small, microscopic. That means this microscopic word means very tiny, small. So they cannot be seen with the naked eye. So what we need to view the cell? We need microscope. You all have seen microscope in biology lab. In next topic, we are going to read about the invention of microscope and the discovery of a cell. The first microscope was constructed by a Dutch scientist, Antony van Leeuwenhoek. So he was an ordinary public official who ground lenses and made microscopic observation as a hobby. He have constructed almost around 400 microscopes. Basically, all his microscopes, they were consisted of a single biconvex lens and they were known as simple microscopes. And those microscopes were having magnifying power of 200 times. Magnifying power means they can enlarge the structure of cell up to 200 times. As you have seen a feature in camera zoom, you can zoom the picture and can view it closely. Similarly, that microscope, they have magnifying power. Here in this picture, one of Leeuwenhoek's microscope is shown here. One is the front side of it, second is the back side of it. This is very simple microscope. So in this microscope, so eye was applied close to the lens. You can see the labeling on one side and object was mounted. Mounted means placed on the needle like screw point on the opposite side of the lens. Next scientist we have Robert Hooke, an English scientist. He developed a microscope by using two lenses. So with the help of two lenses, there was greater magnification. So such microscopes were known as compound microscope. In Hooke's microscope, the object can be seen was placed on the stage below and light from an oil flame was thrown on it by means of concave mirror. Yes, look at this figure 2.2a. So this is the compound microscope, right? So you can see here the labeling is mentioned in blue color. So what is number one? A needle sort of thing is there. So focused on the object below the microscope by a flask. Then second, filled with water, second structure. Then third, fourth, fifth. Third one is eyepiece, fourth one is objective lens, and fifth is tube. So that forms compound microscope. But current microscope is quite different from this. It is actually the improved version of this old Robert Hooke's microscope. So what he did in the beginning, the Hooke examined a thin slice of cork. What is a cork? Woody portion of tree is known as cork. So he observed a thin slice of cork under his microscope, and what he has seen, he saw tiny box-like compartments piled up together. Have you seen honeycomb? It is having several compartments all together. Similarly, he observed such structure. So this is the structure he has observed under his microscope. So these boxes reminded him of the rooms or cells of monk in a monastery. So he said that the cork was made up of cell. The rooms of a jail, they are also known as cell. Why they are known as cell? Because they are small, they are tiny, they are closed. So he actually compared that structure with the structure observed under the microscope. So he named it as cell. So the cell which Hooke saw were all dead cells, and they were empty. It was having only walls. Here it is a structure of ordinary compound microscope. This you have already seen in your biology lab. Further, the invention of electron microscope. Electron microscope is such microscope in which we don't use light to observe the specimen or cell. An electron beam is used to observe the specimen. So these are quite advanced microscopes, right? So they can magnify the structure up to 2 lakh times. A normal microscope, it can magnify up to 2000 times only. The ordinary compound microscope, it uses light, underline this, so which is actually bent by glass lenses to magnify the image. But in electron microscope, what is used? Beam of electron, underline this whole portion. So this is the structure of electron microscope. These are quite expensive. Whenever you get a chance to move to some university for higher studies, there you will definitely find these microscopes. Next, progress check is given. This you have to finish on book only.